This is our first lecture in our discussion of the brain. Our objectives here are to understand how the brain is organized, protected, and supplied with blood, to compare and contrast the various areas of the brain and their functions, and discuss the functional organization of the brain. We will take a look at the brain and the different parts of the brain and some vocabulary associated with the brain. Then we are going to discuss the meninges, or protective fibrous coverings that surround the brain, the ventricles, spaces within the brain, cerebrospinal fluid that fills the ventricles, and the blood supply to the brain. From there, we'll move on to structures in the hindbrain and midbrain, and then we'll move into the forebrain, including the cerebral cortex and how that functions. Some directional terms and landmarks you should know are rostral, meaning towards the forehead, and caudal, meaning towards the cord. The major parts of the brain are the cerebrum, the outer part that looks much like a big blob of thick noodles, so this is our cerebrum. The cerebrum has ridges called gyri and grooves that we call sulci. Another major part of the brain is the cerebellum, which has much thinner gyri. It looks like it's kind of striped or hair-like. The cerebrum is about 83% of the brain's volume, but the cerebellum contains over half of the neurons in the brain. The brain weighs about 3 to 3.5 pounds. The major lobes of the brain include the frontal lobe, which is underneath the frontal bone, the parietal lobe, which is underneath the parietal bone, the occipital lobe, underneath the occipital bone, and the temporal lobe, which is mainly under the temporal bone. These are all part of the cerebrum. In these lobes of the cerebrum, the top 2 to 3 millimeters of tissue is more gray in color because it includes cell bodies and dendrites of neurons. This outer layer is called the cortex. This cortex is made up of gray matter. The grooves we see in the brain, in the cerebrum, are called sulci, or just one is called a sulcus. These ridges we see are called gyri, or just one is called a gyrus. These are helpful vocabulary words as you explore the brain. A couple of other landmarks that are important are the precentral gyrus, the one in front of the central sulcus. The central sulcus divides the frontal lobe from the parietal lobe and goes all the way down from the top of the brain all the way down to this lateral sulcus between the temporal lobe and the frontal and parietal lobes. The postcentral gyrus is just behind the central sulcus. Gray matter in the brain includes neuron cell bodies, dendrites, and synapses. This gray matter forms a cortex over both the cerebrum and the cerebellum. Gray matter is also found in nuclei deep within the brain. When we discuss the brain and we call something a nuclei or nucleus, it's a group of cells that have a similar function. So they work together to control something. This is not the same as the nucleus of a cell. So in the brain, a nucleus is a group of cells that work together. White matter is made up of bundles of axons. These bundles of axons are connecting different parts of the brain. They may connect different lobes or different areas within a lobe or even different hemispheres of the brain. These bundles of axons are called tracts. The meninges are protective membranes that surround the central nervous system. The dura mater note the spelling with one T, is the outermost tough membrane. It has two layers, an outer periosteal layer that's directly against the bone, and that's separated from the inner meningeal layer to form our dural venous sinus. This is one of the areas that drains blood from the brain. The dura mater also forms some supportive structures that help keep the brain in place and keep it from moving around within the skull. As we go down the back, the dura mater continues to surround the spinal cord as well, and there's an epidural space, a space just superficial to the dura mater, that's filled with fat in the low back. This is where epidural anesthesia is applied during childbirth. Here you can see that we have our dura mater, which is just underneath our bone, and there are two layers that make up the dura mater. There's the periosteal layer against the bone and the meningeal layer that's slightly deep to that. You can see here that this triangle between those two layers, the sinus, is being used to drain blood from the brain. Our next layer of the meninges is the arachnoid mater. This arachnoid mater has small processes that stick down that look kind of like spider webs. There is a space right underneath the arachnoid mater called the subarachnoid space. It's filled with blood vessels and cerebrospinal fluid. The pia mater is the protective layer directly on top of the brain, and it adheres to the cerebral cortex. Underneath our dura mater, we saw our subdural space, and it's a very narrow fluid-filled space. Under that, we have our arachnoid mater, which is a thin membrane loosely covering the brain that forms a subarachnoid space, which is a wide space between the arachnoid mater and pia mater. This space contains cerebrospinal fluid and blood vessels. The pia mater is a thin membrane that directly adheres to the brain. Meningitis is a disease of the meninges. It's when the meninges become inflamed. This can be due to either bacterial or viral causes, and it's generally a disease of infancy and childhood, between about 3 months and 2 years of age. These bacteria and viruses can invade the central nervous system by way of the nose and throat, and signs are going to include a high fever, stiff neck, drowsiness, and an intense headache that may progress to coma if the meninges are so inflamed that they compress the brain inside of the skull to the degree it causes coma. This is diagnosed by examining the cerebrospinal fluid via a lumbar puncture. So in the cerebrospinal fluid, either bacteria or a virus should be found, and that can determine what type of meningitis the individual has. Here's what a lumbar puncture looks like. They take a needle and go between two of the spinous processes of the vertebra. So right in here, here's our needle. So we have the spinous process of L4, the spinous process of L5. We sneak right in between them and stick the needle all the way into the meninges of the vertebral column. Then a small amount of cerebrospinal fluid is removed and then examined. Next, we're going to take a look at how this cerebrospinal fluid is formed.